Okay, so major group of parasites. Helminths, as I said, they include nematode, trematode, and cystode. Protozoas, arthropods that include insecta and arachnidas. So why we bother about the life cycle or taxonomy or the biology of these parasites? Because these parasites have a great similarity as far as the biology life cycle is concerned, and they are very closely related to each other. Their eggs or their adults are also very closely related to each other, are very similar. Therefore, the knowledge of these particular parasite biology or life cycle will help to understand the pathology of the disease, clinical features of the disease, how you can diagnose these diseases, and what the treatment plan as well as control strategy you will be adapt for these parasites. So these parasites are highly successful organisms. There's a lot of parasites in the world, plant, animal, and but these parasites, the beauty of these parasites are they have a high level of genetic diversity. For example, this high level of genetic diversity in these parasites will helpful to adapt them in a number of different environments. For example, I will not give you example of plants or some other human parasite. I'll give you example of animal parasite where your interest is. We will talk about some of the parasite like Haemonchus. Is a very common parasite of livestock in the number of in different lecture. But this parasite is a tropical parasite. However, nowadays this parasite is now seen in Canada, in a highly temperate region, in Sweden, in Scotland. So I'm trying to establish here how this genetic diversity make these parasites to be adapt in number of different environment. Also, this genetic diversity in these parasites create a big problem of drug resistance, because the mutations in their genes will be, due to the genetic diversity, will be very high, and they cause resistance against these drugs. So that's why these are really successful organisms in the earth. So a relationship between host and parasite. Many parasites are quite host specific. Some has some zoonotic importance, but like horse or like livestock parasite, they are very specific to these species. Dogs parasite, they are very specific to them. Lice are very specific to the host. Mites are fairly specific, but they also cause some switch their host, like some of the mites will cover in this term that they are also cause infection in human as well as animals. Direct host to host transmission is rare, especially in the internal parasites. So these parasites do not directly cause mortality. In very few of them cause the mortality, especially the Helminth parasite. They cause a production losses. Right? They cause reduce the productivity of the animal. They cause chronic diseases. So the Helminth parasite, they modulate the host immune system, and they can also suppress the Th1 type cytokinase pro-inflammatory arms of the immune system. So these are the number of different relationship between the host and parasite. So all parasites are not treated equally. Yes, we will focus on these species that are clinically relevant for you as a vet. So why are parasites important? This is one of the reasons these parasites are important. These are some of the parasites. We will be covered all of these parasites in a number of different lectures. In dogs, these Toxocara, as well as if you look into this ported belly is caused by hookworm, encyclostoma. And in horses, if you look, these are Parascarisquarum. These cause a very severe disease in horses. And this parasite is Homuncus, causes severe disease in sheep and goat and cattle. So that has a very severe effect on the health and welfare of animals. So as far as these Helminth infection in animal, the puppies, these are born with these worms. For example, hookworms. You will see these worms they will be transferred via the placenta. These Toxocara. These are transferred by placenta, and through milk. Hookworm is normally transmitted to the puppies from after the birth. And in the grazing animals, the story is a little bit different. You will see these larval stages in the grass when the ruminant will be going to the grass and do the grazing. So they will be taken these infective stage larvae, and they complete their life cycle. So this is how the infection can be transmitted in a number of different ways from one generation to the other generation. So these are some of the examples of parasitic infection in human. So this is Brugia malayi. This infection this parasite is very similar to heartworm parasite and is exactly the same filarial superfamily and caused by mosquito. Also, you will see the encyclostoma, that is hookworm. Hookworm infection is also common in human, and this is dracunculus, is a guinea skin worm. So that is also common in human. Okay, so anyone has any question before we can move on? Okay, so for you as a veterinarian, treatment and control strategy is really important for your practice income. And we'll deliver some of the lectures in this term that will give you the best idea how you can control these parasites. For example, Ray will be covered heartworm control strategies. We'll also cover some zoonotic parasite control strategies and also flea and tick control strategies. So these control strategies will give you a learning how you can educate the client once you go into the clinics. And also we'll be talk about some control strategies for the livestock parasite. That has a very strong impact on the production of these livestock, because farmers always depends on the production of the livestock, and if the parasite control strategies will not there, the production will low, and that will not useful for the farmers. So here, good worm control strategies will increase the weight gain for the farmer. If the weight gain will be increased, they will get a good income, they will be happy. So this is what you have to teach, educate these clients, that how you can increase the productivity of your animals. Okay, so quickly we can go through some of the definition of what is parasite. So there's a number of different definitions here. And I always like this animal living inside of the other animal. So I always like this definition of parasite. So life cycle. What is life cycle? So this is the life history of the parasite. So in what particular time point parasite complete their cycle to become adult. And this is important because if you don't understand the life history of the parasite, you can't able to do the control strategies, you can't able to do the treatment strategies, or you can't understand the pathogenesis or clinical features of these diseases. So that's why the life cycle understanding of the life cycle is very important, and you don't need to memorize it. It's a basic understanding we will deliver a number of different lectures where you can see how these life cycle you can understand in a different ways. Okay, so type of life cycle. Direct life cycle mean there is a one single host. The life cycle will be complete in a one single host, and some of the stages are the free living stages, and they are in the environment or in the grass. And once they converted into the infective stage, they can eaten by the animal from the grass, and then the rest of the stage will be complete in the predilection site or in the abomasum or in the rumen or is in the intestine. Then the life cycle get completed with a single host. Indirect life cycle, where the more than one host will be involved. For example, trematodes, they have an intermediate host. Snails are the intermediate host, and the half of the life cycle complete in the intermediate host, and then they can shed this one of the stage in the grass, and then they can further be taken by the definitive host, and the life cycle get completed in the definitive host. So definitive host is a normal host in which the parasite reach into their sexual maturity and reproduction occur. This is how the definition of a definitive host is. And what about the intermediate host? The parasite grows but not reach into the stage of sexual maturity. But the intermediate host is required for the life cycle to be get completed. Without the intermediate host, life cycle will never be complete. So what is the difference between intermediate host and the vector? Vector is also intermediate host, like vector is arthropods. These are the vector of some of the protozoan diseases. But in the intermediate host, they can the life stages will be converted, and this one of the particular stage will come out from the intermediate host and go into the grass and taken by the definitive host. But in this case, arthropod, because they are like ticks, for example, they can take these one of the stage from the definitive host through blood and transmit into the other host. So this is the difference between the intermediate and the vector. You should understand the difference between what is the intermediate host and what is the vector of any particular disease. Biological vector or mechanical vector. Biological vector is the vector where the sexual reproduction has been happened within the tick, and let's say example as a tick or fly, and the asexual reproduction happen in the definitive host. In the case of the mechanical vector, there is no sexual reproduction happen in the vector. They just take the blood and the parasite and just transmit into the other definitive host. That's it. This is called a mechanical vector. No transmission happen. Okay, so paratonic are transport hosts. These transport hosts, they are not a part of the life cycle, but they are just a helper to take the intermediate host to the definitive host. So they are just like a transporter. It's not more than that. Aberrant or accidental host. So sometime what happen, intermediate host, they get the disease and they further convert it into the number of different stages, but sometime some host, they accidentally infected by the disease, and they are not normal host or definitive host of that disease, but this is accidentally happen, and normally the pathology of the disease is not occur in that accidental host. However, sometime we have seen that some of the disease show a very severe clinical sign in the accidental host, but most of the time they are just a dead end hosts. Pre-patent period. So this is the period when the definitive host take the infected stage of the parasite and they go into the predilection site. For example, predilection site is small intestine. They go there, and the time from that particular time to the time where they show up the clinical features, that is the pre-patent period. So it is the time before the potency of the disease or severity of the disease will be start. So this is really important to understand because without knowing that once the disease clinical signs comes up, is important to be further diagnose the disease, or you should go with the treatment plans or the control strategies. So this is how we define the pre-patent period. So anyone has any question? Anything if you in your mind related to the course, what it should be look like? If you have any confusion, just raise your hand and ask. Everyone is clear? Okay, great. Thank you.